Hello everyone, my name's Paul Maruff. I'm Chief Scientific Officer at Cogstate, and in this video today what I'm going to do is take you through and introduce you to the way in which we use cognitive tests to make decisions about drugs in clinical trials. So what I want to cover is three main things when we're thinking about how you assess cognition. First of all, why do you assess cognition? What do cognitive tests actually measure and how they are used in a clinical trial? And it's probably worth, even before we begin, considering what the word cognitive actually means. So cognitive is another word for thinking, and a cognitive assessment is essentially an assessment of thinking. And largely we use these assessments to allow sponsors or researchers to make decisions about the extent to which the drugs they are studying is affecting or are affecting the way in which people think. Now, the important part about that is that if the drugs are influencing or affecting the way people think, then they're acting on the central nervous system. That could be a good thing or that could be a bad thing, but either way, it's an important thing to know. What I'm going to do is I'm going to illustrate with a couple of case studies just different dimensions of that idea about how we use cognitive tests to tell us about the ways in which the drugs are either facilitating or somehow inhibiting cognition. So if you look at this slide here, what you're seeing is a summary of data from a cognitive test where an individual did that cognitive test at some time when they came to the laboratory. The drug was given, and then in order to measure the acute effects of that drug and how thinking changed with time after administration, you can see that the test was reapplied here at 40 minutes after dosing, 2 hours after dosing, 4 hours after dosing, and 6 hours after dosing. There are two conditions here: a group of people who received placebo, indicated by the dotted line, and a group of people who received the drug, indicated by the solid line. And it's pretty clear what's occurred here. Things have changed immediately after the drug was given, but as the time from dose increases, the changes in thinking or the effect on thinking diminishes, so that by 4 and 6 hours, individuals who have had the drug and individuals who haven't had the drug, you could say, are thinking at the same level. But it's useful to consider some of the aspects of this data. The drug is esketamine, and this is data from part of the phase 2 program, and ultimately this drug went on to be approved by regulators for the treatment of treatment-resistant depression. Now, esketamine is an NMDA modulator, and one of the things that the company was worried about or concerned about would be that the doses at which esketamine was going to be used clinically may cause individuals to become somnolent, to lose their ability to concentrate or make decisions, or to cause them to lose their memory or the like, and that effect might be abiding. And what this figure shows is performance on a test called the Groton-Maze learning test. And the unit of measurement on the y-axis is the number of errors people are making on that test. And you can see that prior to drug, the individuals made about 35 errors, both those in the placebo group and those in the active group. After the drug, the people in the active group, the number of errors increased to about 60. However, those in the placebo group, errors remained relatively stable right across the whole study day. So a first and important part of this maze test, you can see, is that its reapplication wasn't associated with any changes in performance by itself. So people didn't get better at doing the test for having done it, or they didn't get worse at doing the test for having done it. But rather the test could be given repeatedly without eliciting any behaviour change. That means then that we can see and make an inference that the increase in errors that occurred immediately after the dose of esketamine at 84 milligrams reflected the effects of that drug on the central nervous system, and that was quite a substantial change. However, this data shows you that by 2 hours after dosing, the number of errors people were making on that test had reduced substantially, almost back to baseline. And while those points there at the 2 hour are not statistically significantly different, you can get a sense that performance is still returning to normal. But you can see at the 4 hour and the 6 hour assessment that the number of errors is exactly the same in both groups. So our ability to apply this test repeatedly meant that we could compare performance before and after drug and make decisions, first of all, about the extent to which the drug influenced this aspect of thinking, and second, about the timing of that influence and for how long it remained. So what we're able to see from this data is, yes, there were some acute negative effects of the drug on maze learning. Your ability, and here maze learning reflects your ability to integrate information, to make complex decisions, to solve problems. But after 2 hours, that had returned to normal and remained normal thereafter. So this provided for us a basis for us to make decisions about issues like how long we need to look after people after we've dosed them. It suggests that 40 minutes after dosing, we need to really have people in a safe and controlled environment. But 1 to 2, or 2 hours later, people are essentially making decisions as well as they were making prior to their taking the drug, and that there's no abiding or lasting effects of that drug on their decision-making ability. Considering this data on the esketamine challenge with the maze brings up a couple of issues, right? What are the characteristics of the cognitive test that we've given here that would allow it to be used in this manner to guide those sorts of decisions that I was just discussing? And the points are, first of all, we require that the test be valid. And by valid, we mean that the test measures what we think it's measuring. The Groton Maze Learning Test is a test, as I said, of decision-making, problem-solving, and in a neuropsychological context is called executive function. So this is established for this test. You can see that the retests occur relatively quickly. 40 minutes after dosing, 2 hours after dosing, 4 hours after dosing. So in this context, it's crucial that the test itself not take up lots and lots of time. In fact, the less time it requires, the better. The next aspect of this test that's important is that it is repeatable and does not cause individuals' performance to improve as a consequence of that re-administration. This is evident in the performance under the placebo condition given by the dotted line, where it can be seen that the number of errors made with re-administration remains relatively stable at about 33 or 34 errors right across every visit, despite the fact that individuals have not received drug and they're doing the test multiple times. When we designed this study and recommended this test to answer this question, we did so because we had established that this test is itself sensitive to change. And that is that we need to ensure that the test can detect changes in thinking. Now, in this case, we can see that it in fact did, and that the performance declined as a consequence of the dosing in the early stages. But our ability to infer or to conclude that there was no ongoing or abiding effects of the drug at the later visit requires us to understand and trust that that absence of change, that the absence of difference reflects that truly there was no difference. So we have to show that the test is sensitive to change when it truly occurs, and that performance on the test won't change when there's been no change to central nervous system function. Finally, an important aspect of the test for its use in clinical trials is that there are no floor or ceiling effects associated with its use. So you can see that in this study, despite the fact that individuals were healthy, that they still made a number of errors to begin with. And you can see in the placebo group that despite their repeated testing, performance never got any better. So we got to no ceiling. People didn't get to the maximum. They weren't able to perform at the maximum values of the test, despite the repeated the repeated application of the test. So that is, the test has no ceiling effect. And you could see, for example, if we were asking questions about the extent to which a drug could improve performance, a ceiling effect might diminish our ability to see that. In this case, though, we're interested in the extent to which a drug can impair performance. And there the issue is that we want to make sure that the number of errors is such that individuals don't reach a minimum value or a floor effect. So on the maze, you can make many, many hundreds of errors here. People, the healthy folks, after they'd been administered the drug, made 60 errors, right? But they were still a ways from the maximum number of errors that could be made. That value was still substantially less than the maximum number of errors that could be made on this test. So we had sensitivity to detect that decline and to characterize that decline. So these dimensions of the tests together bode well for its application in these type of studies that allow us to make the decision about the safety here or potentially the efficacy of the medicines. This slide here just shows you a cartoon of what the Groton Maze learning test looks like if you're actually doing it yourself. And largely it's a 10 by 10 matrix of tiles, and hidden under those tiles is a pathway. And the individuals must select the tiles one at a time to try and find where that pathway exists. After they've found the pathway, the next step of the pathway, it vanishes behind them. So they have to build up a representation of where that pathway is in their memory. They do that a number of times, 5 times. And with each time, each trial in which they solve the problem, the representation of the memory becomes stronger and their performance improves. But you'll recall from the slide I just showed you that individuals doing this test repeatedly got no better at it, right? So they're learning to solve and improve their performance within a session, but across sessions, there's no improvement. And the reason why that occurs is that on each session, we get them to learn a different pathway. And so this provides for us a great opportunity to measure the way in which individuals can solve problems. While we're on the maze effect, it's useful to consider here with the same test, the way in which individuals' perceptions of their cognition and individuals' actual cognition can be quite different. I think this is illustrated nicely in this publication by Cromer et al., where the maze was applied in a relatively simple study where it was measured repeatedly as individuals' blood alcohol concentration increased. And on the left figure, you can see that individuals' perception of their sensation of intoxication increases linearly with their blood alcohol concentration given on the x-axis as their blood alcohol level is ascending. And then as the alcohol reaches its maximum and then starts to decrease, you can see that their sensation of intoxication also decreases. But you can sort of see there's a little asymmetry in that the rate at which it's decreasing or the sensation of the decrease is a little different to the increase. If you look at the middle figure, that's that same data superimposed as an ascending line and a descending line as a function of blood alcohol concentration. And what you can see is that individuals are rating themselves as more highly intoxicated as their blood alcohol concentration is ascending. And that for the same dose of blood alcohol, when on the descending part of the curve, you can see that they're rating themselves as being less intoxicated. So their subjective sense of intoxication is changing as a function of whether blood alcohol is increasing or decreasing. On the far right figure is performance on the maze through those same time points. And there you can see that the number of errors people are making is related to their blood alcohol concentration, irrespective of whether the blood alcohol is ascending or descending. So whilst people are feeling less intoxicated as their blood alcohol is descending, in actuality, the level of cognition is impaired on the descending arm of the curve as it is the ascending arm of the curve, and is related to the concentration of the drug, despite the fact that individuals don't feel as impaired at that same time point. So what this shows us is that individuals' subjective sense of intoxication, and this can extend as well to subjective senses of euphoria or subjective senses of the ability to concentrate, often don't map completely onto what occurs with an objective assessment of cognition. Now, this doesn't mean that one is better than the other. It just means that these different sources of information are telling us different aspects about the way in which these drugs, or in this case alcohol, can influence central nervous system function, and that it influences the objective, or it influences thinking in a different way than it influences the way individuals think about their thinking. Both of those are very important, but they tell us different aspects of the drug effects. So now we can use information that we learned about the effect of alcohol on complex decision making, that is, the effect of alcohol on performance on the Groton-Mays learning test, to help us understand the meaning of the effect of esketamine on the Groton-Mays learning test. So one of the things that we know is that in many jurisdictions, blood alcohol concentration is used as a criterion by which we make decisions as to whether people should drive or not. And whilst the value that people use to make that criterion differ in different countries and in different states, it's common. A common criterion is 0.08. If you think about that, that's sort of central nervous system legislation in a way. Really, the idea is that with that sort of blood alcohol concentration, your ability to make decisions and guide purposive behaviour is diminished to such an extent that we would prefer that you weren't driving a car and putting the rest of us at some risk. So if we take that as an example, you can see in the figure on the left that individuals started about 40 errors at blood alcohol concentration of 0.02, and then that number of errors increased to about 48 errors at blood alcohol concentration at 0.08. And so here we've had an increase of about 8 errors. So that information helps us understand the effect that we observed in the esketamine. In esketamine, at baseline, individuals were making about 35, 34 errors before the drug, and after drug, that increased to 61. So the effect of esketamine on the number of errors, or the effect of esketamine on purposive behaviour as measured by the Mays, was much greater than the effect of alcohol at the level of 0.08, that level that as a society we would prefer that you weren't driving. And so we can start to understand aspects then of the importance of the effect. Another way to conceptualize that would be to express it as a magnitude of effect size, which is the difference in errors as a function of the pooled variance. Now, we could go into that in another context, but as a measure of effect size, 0.08 gives us an effect size of 0.8, D equals 0.8 there on the figure. And by comparison, you can see that the effect size for the esketamine is much greater, almost twice as large. So we can help now to make decisions not just about the presence of the abnormality that occurs as a consequence of acute dosing with esketamine, but we can also talk about the magnitude and the importance. And we can make statements like, that's an important impairment, and potentially even further, that this should give us some concern about our willingness to allow people to drive or to make important decisions immediately after dosing. Now, of course, this is not a driving test. What we're using here is really an idea, or a context by which we can communicate and understand the importance of any changes that we observe as a consequence of the administration of a drug that we don't understand well. Making use of the idea that we have another drug that we do understand quite well, and in fact many people understand quite well, which is the effects of alcohol on your ability to make decisions. Okay, so we can push on there. We talked a little about the maze, and on this slide now what you're seeing is in a different context, looking at the effect of the maze on an experimental drug that was being developed by AstraZeneca. Importantly though, what I'm going to focus on is what was used as an active control in this study, which is a drug called lorazepam, given at 2 milligrams, which is the therapeutic dose. And what you can see on the left figure, again, is the GMLT, the Groton Maze Learning Test errors. And you can see that, like these other two studies, that healthy young adults prior to drug make about 40 errors on that test. And you can see that after lorazepam 2 milligrams, 2 hours later, they were making about 80 errors or 78 errors here. Now, unlike esketamine, you can see that the effect of the drug lasts much longer. And in fact, performance or the number of errors on that maze doesn't return to normal levels until about 8 hours after dosing. So if we come back to the context that we looked at before, the number of errors increases from 40 to 78. So we know that was bigger than what we observed with alcohol at the legal driving limit. And to reconceptualize that as an effect size, you can see that the effect size there is 1.21. So the effect size is not as great as that which we observed for esketamine, but it's certainly more than that we observed for alcohol. And the other thing we can say is that unlike esketamine, that large deterioration that occurs 1 and 2 hours after dosing remains present up to 8 hours after dosing. And in fact, even at 4 hours after dosing, the people are still making 70 errors, an increase greater than what we saw with the alcohol. So it helps us understand the nature, the magnitude, and the timing. And again, if you look at that figure, at the experimental drug at each dose, by comparison, you can see that that had no effect on this maze learning test. So if, as the company was investigating at the time, this drug was aimed to be used as an anxiolytic drug, then one characteristic of that is optimal, or that appears to be becoming optimal in early phase, is that the drug itself has no effects on cognition. That is, no deleterious effects on cognition at either dose. So the decision that we can make here is that that's a good characteristic of the drug. Another important thing is that the placebo condition in this study is itself, again, not changing as a consequence of re-administration. So therefore, the absence of any change in the active experimental drugs, we can make with great confidence that that reflects that the drug itself was having no influence on this effect, on this aspect of thinking, right? On this more higher level aspect of thinking. The figure on the right illustrates the effect of this same drug on a different aspect of thinking. Here the test is psychomotor function, and psychomotor function is measured by the detection task from the Cogstate brief battery. And essentially the detection task is a measure of simple reaction time. And you can see, like for the maze, that simple reaction time under lorazepam 2 milligrams diminishes substantially immediately after dosing. And you have that same profile where the impairment remains until 8 hours after dosing, where it returns to normal limits. But if we conceptualize or reconceptualize that data, the change that we observed in simple reaction time from baseline to 2 hours after dosing, you can see that the effect size there is 1.73. So that's much greater than that which we observed for the maze, which is shown on the figure on the left. So we could make a statement there that whilst higher cognitive functions are affected, and affected in a substantial way, reflexive or automatic aspects of cognition, that is your ability to concentrate and attend, are affected to an even greater extent by this drug. Although, like the higher aspects of cognition, the characteristics of the change in cognition are the same in that they reach a maximum at about 2 hours after dosing and then return to normal limits about 8 hours after dosing. An interesting aspect of performance here, though, is you can see that the higher dose, the 40 milligram dose of AZD6280 itself is associated with some decline in cognition, small but present. And so we can use that then to actually make decisions about dosing. This early phase study, we could say that if a characteristic of this drug that was a necessary condition for it to move forward was that it identified no deleterious cognitive effects, we can see at the higher dose at 40 milligrams, there is some deleterious cognitive effects, and we can actually start to understand or make decisions about whether they're important or not. But we certainly have some information here to help us with that. A second important part too is that the fact that there's a negative effect illustrates that the drug is getting into the central nervous system and in sufficient amounts that can interfere with normal cognitive function. And then we can go back and say that whilst it gets into the central nervous system at that dose and interferes with concentration and rapid decision making, when we look at higher level cognition, purposive behavior and problem solving, it doesn't affect that. So we can start to characterize and quantify the way in which drugs affect different aspects of cognition differently at different doses. And this information can be integrated as part of drug programs to understand the characteristics of the drug and where and when it might be dosed or how it might be dosed and what sort of things we can expect in patient groups.